In this tutorial, we will learn the quantitative analysis of skeletal muscle fiber area using ImageJ software. Here is an image of a TA muscle cross-section stained with green, red, and blue. Today in this tutorial, we will measure the area enclosed by the laminin staining that corresponds to the skeletal muscle fiber area. Since our interest is to measure the area enclosed by the laminin, therefore, we will use only the green channel and disregard the blue and the red channel. To achieve this, let us split the colors into 3 different colors. To do this, drag and drop the image of interest into ImageJ and click on Image, Color, and Split Channels. At this point, we can disregard the blue and the red channel. You can also increase or decrease the brightness and contrast according to your choice. During this process, the pixel might be changed. Following this, the brightness and the contrast window can be closed safely. Make sure that the image is a 8-bit image. From the image, it is evident that there are gaps that exist that prevents the complete enclosure of the laminin staining. To use ImageJ software for area analysis, these gaps may be closed. To do this, click on the color picker from the tools above and select the color of your choice. Here in this image, one can click on the white spot, and once the color is picked, click on the brush tool from above and start to fill the gap that exists in the skeletal muscle fiber cross sections. If the gaps are left unattended, then the software may fail to calculate the specific area specified for the particular fiber area. Once the gaps are filled using this method, the image is ready for further processing. The next step is to adjust the threshold level. To do this, click on Image, Adjust, and Threshold. To calculate the area, the default option can be selected from the drop-down, and in the color, the red color can be selected. You can also select the dark background and do not reset range. To adjust the threshold, the 2 sliders needs to be used. The first slider can be adjusted to all the way to 0, and the second slider needs to be adjusted according to the image. For instance, the second slider can be kept at 37 for this image. The threshold is set such a way that the enclosed area is colored red, while the gap between the enclosed area is marked as white. Once the threshold is set satisfactorily, click on Apply. The next step is to convert the image to mask. To do this, click on Process, Binary, and Convert to Mask. Following this, the image needs to be eroded. Click on Process, Binary, and Erode. At this point, the edges seem to be smoother. Before calculating the area, the final step is to invert the image. To do this, click on Edit and Invert. Upon inverting the image, the foreground, that is, the area enclosed by the laminin, is white in color with a black background. Now, the area is ready to be measured. Click on Analyze and Analyze Particle. It is very important to define the size in pixel square. The minimum size can be adjusted according to the type of the muscle fiber area. For this image, we will use 1000 as a lower end and 900,000 as a upper end. The circularity can be kept as it is. In the next drop-down, show select Overlay Mask. And from the options below, check on Display Results, Summarize, Exclude on Edges, and Overlay. Once the parameters are set, click on OK. Upon clicking OK, a bunch of results display. Let us discuss one after the another. Now, the muscle fiber area enclosed by laminin staining has been indicated by different colors. Within the colors, there are specific numbers. These numbers are assigned by the ImageJ software as an identifier to their specific corresponding area. In the summary window, it shows the total count as 31. This indicates that there are 31 muscle fiber cross-section area in this image, and each fiber area is assigned a different number. In the results window, it displays all the 31 muscle fiber area corresponding to their specific numbers. For instance, in this image, the red color coded fiber is assigned as 1. From the results section, it is evident that this fiber area corresponds to an area of 3837 pixels square. Similarly, the fiber area of each of the individual fibers can be identified using this method. The results section displays all the 31 muscle fiber area. One can select all the fiber area and copy it to an Excel file, or it can be directly saved as an Excel file. Now, the data from the Excel file can be easily used to generate a graph showing the quantitation of the muscle fiber area. This image generated by the ImageJ software can be easily saved into a JPEG format. To do this, click on File, Save As, and select JPEG. You can rename the file as you would like to, and then click Save, and the image is saved in a JPEG format. Thanks for watching the tutorial. If you have any questions, please put it in the comments section below.